Greetings listeners, my name is Warot Cholasak. My advisor is Ajahn Rangsimarian Trakul. Today I'm gonna talk about gene expression by nanostring RNA sequencing of myeloid-derived suppressor cells in knee osteoarthritis. The knee joint is a hinge-type synovial joint which permits flexion and extension of the knee. The synovial tissues lining the non-articular joint surfaces are specialized 1 to 3 layers of columnar fibroblast-like synoviocytes intermixed with macrophages. Homeostasis within the synovial tissues depend on many variables, including the production of lubricin, matrix metalloproteinases production, regulation of immunity from various cytokines, growth factors, and white blood cells. These environments are crucial for maintaining the stability of the knee joint. Infrapatellar fat pad or Hoffa's fat pad is located at the anterior knee compartment. It is a mass of adipose tissue which is classified as intracapsular but extrasynovial. This fat pad is believed to be a potential source of inflammatory process in osteoarthritis, as it is found to contain immune cells that can secrete pro-inflammatory cytokines, including interleukin-6 and its soluble receptor. The complete pathogenesis of osteoarthritis is still unclear. The endpoint of OA is gradual degeneration of articular cartilage and other morphological damage to the joint, but the small biochemical and molecular changes have occurred in the beginning. The majority of OA cases have a coherent predisposing condition. The disease is not totally an inevitable age-related degenerative disease. Major risk factors include aging, trauma, obesity, chronic mechanical load, and genetics. As said in the last slide, aging leads to collections of damaged unrepaired DNA, proteins, and lipids from reactive oxygen species and reactive nitrogen species, which can lead to cellular defects and abnormal tissue function, which could result in cellular senescence and abnormal cytokines and chemokines. In terms of trauma, examples can be seen in post-traumatic OA from abnormal joint loading. 39 to 78% of the cases can be accredited to genetic factors. Some of the studied genes can determine the age of the onset, location of the affected joints, and severity and progression rate of the disease. For obesity, mild persistent systemic inflammation, so-called meta-inflammation, can lead to immune dysregulation, which leads to abnormal cytokines, chemokines level, and cellular equilibrium, which can eventually result in inflammation of the joints. And lastly, chronic mechanical loading can lead to OA. Myeloid-derived suppressor cells are certain groups of cells that originate from the myeloid lineage that has immunosuppressive function. They are composed of mainly two cell types, polymorphonuclear MDSCs or PMN-MDSCs and monocytic MDSCs or MMDSCs. PMN-MDSCs display a morphological and phenotypical similarity to neutrophils, while MMDSCs are comparable to monocytes. Both are present in a very low percentage in a normal individual peripheral blood. However, a considerable higher amount of both cells are observed in many diseases and conditions, including various cancers, chronic infection, autoimmune disorders, obesity, and pregnancy. Chronic inflammation and infectious disease induce a comparably longer period of weak immune activation, triggered mainly by inflammatory cytokines including interleukin-1 beta, interleukin-6, GM-CSF, and MCSF, and endoplasmic reticulum stress response. This long and weak activation leads to poor phagocytosis, constant production of ROS and NO, arginase 1, anti-inflammatory cytokines, and prostaglandin E2. These can disturb the homeostasis of cells, resulting in cell death and apoptosis. This table compares neutrophils, monocytes to PMN MDSCs and MMDSCs respectively. The activation stimuli differs. Short duration of activation stimulates neutrophils and monocytes, while longer and chronic exposure to cytokines leads to activation of MDSCs. Cell surface markers are different. You can see the main regulators of suppressive functions of MDSCs such as STAT1, STAT3, and STAT6, NF-kappa B, and prostaglandin E synthase, etc. The mechanisms of immunosuppressive function of MDSCs in a variety of disease are listed in this slide. There are 8 main pathways of suppressing the immune system. The first mechanism is the production of prostaglandin E2. Prostaglandin E2 can bind to 4 different G protein couple receptors, which are EP1 to 4 receptors. EP4 is specifically highly upregulated in a multiple variety of cancers, including lung, ovarian, breast, and prostate. The production of prostaglandin E2 can inhibit the effector T cell, dendritic cells, and NK cells. It can also increase the regulatory T cell response, survival, and proliferation. And it can also increase the MDSC itself using autocrine signaling. The second mechanism is amino acid depletion and the production of immunosuppressive metabolites. MDSCs can produce large amount of intracellular arginase, which could escalate the uptake of environmental arginine, leading to a depletion of arginine, which is vital for the effector T cell, mainly cytotoxic lymphocytes, to function properly. MDSCs can also uptake a large amount of L-cysteine, which has similar effects on T cells. The third mechanism is reactive oxygen species and reactive nitrogen species. PMN MDSCs showed a great amount of ROS production but little nitric oxide due to augmented expression of STAT3 and NADPH oxidase, whereas MMDSCs displayed high levels of NO release but low ROS activity, owing to increased expression of STAT1 and iNOS. ROS can inhibit proliferation and promotes apoptosis of T cells, while RNS can inhibit IL-2 signaling. The fourth mechanism is direct engagement of T cell inhibitory and apoptotic receptors. The first is galectin-9 TIM3. GAL9 can bind with TIM3 on T helper 1 cells and inhibits cellular response and also induces CD4 and CD8 T cell apoptosis. MDSCs are found to be able to secrete interferon gamma, which upregulates the expression of both TIM3 and GAL9. The second is Fas ligand and Fas. Using the caspase pathway, MDSCs can express Fas ligand, which can promote immune suppression from T cell apoptosis. Third is PD-1 and PD-L1, which is an immune checkpoint. MDSCs express PD-1, which can lead to an activation of T cells. The fifth mechanism is the production of inhibitory cytokines by MDSCs, mainly interleukin-10. MMDSCs are found to be able to synthesize interleukin-10 in cancer patients. IL-10 has the ability to suppress both proliferation and cytokine production in multiple types of cells, including effector T cells, CD4 T cells, and CD8 T cells. Interleukin-2, interleukin-12, and interferon gamma production are also decreased by the cytokine effect. Moreover, interleukin-10 could induce expression of PD-1 with STAT3 dependent mechanism to inhibit immune response by T cells. The sixth mechanism is MDSC-mediated inhibition of T cell homing to the lymph nodes. They express proteins including ADAM17 and disintegrin, which possess the ability to cleave L-selectin CD62 ligand on naive T cells. The seventh mechanism is production of extracellular adenosine by MDSCs. Adenosine exerts its function via binding to receptors A2A and A2B on a variety of immune cells, leading to immunosuppression and cancer survival. And the last mechanism is S100A8, A9, mediated accumulation of MDSCs. The cells themselves secrete S100A8 and S100A9, which involve in MDSC's reproduction and recruitment. There's a correlation between obesity, osteoarthritis, and MDSCs. High-fat diet promotes MDSC's expansion in peripheral blood of OA mice and human subjects, and osteoclast differentiation in mice, leading to subchondral bone destruction, which leads to osteoarthritis progression. MDSCs can be reprogrammed into osteoclasts with the presence of MCSF and RANKL. Moreover, MDSCs are found to be present in the joint of OA patients as well. This figure shows that the high BMI is associated with a great number of MDSCs. Both PMN MDSCs and MMDSCs are present in the peripheral blood and synovial fluid of OA patients, with likely predomination in obese group, although statistically insignificant. Our research design is an observational or analytical study. The research methodology. Our population is knee osteoarthritic patients in King Chulalongkorn Memorial Hospital. Our target population is osteoarthritic patients in King Chulalongkorn Memorial Hospital who undergo total knee replacement surgery. Synovial tissues and infrapatellar fat pad from the knee of osteoarthritic patients are collected between April 2022 to December 2022. Our inclusion criteria include a patient that is diagnosed with knee osteoarthritis, patient that is going to undergo knee arthroplasty for knee osteoarthritis, and age 40 to 90. Our exclusion criteria is osteoarthritic patients with other inflammatory knee joint conditions, including rheumatoid arthritis. For our method in identification and analysis of MDSCs using flow cytometry, osteoarthritic patients will be recruited and specimen collection will be done by the Department of Orthopedics. The samples will be collected, including synovial tissues and infrapatellar fat pads. The samples will be pooled together within the same group. There are two groups of patients. The first is obese and the second being non-obese. Obese is defined by BMI more than 25 and non-obese by BMI less than 25. Then MDSCs will be identified using flow cytometry and analysis of MDSCs between the groups. This is our preliminary data which shows that MMDSCs are present in the knee joint of osteoarthritic patients. This is identified by specific pattern of cell surface markers expression, including negative in HLA-DR, but positive in CD68 and CD14 as shown in the picture done by flow cytometry. The next step is gene expression analysis of MDSCs using nanostring technology. The first step is isolation of MDSCs using fluorescence activated cell sorting or FACS. Next is RNA extraction using RNase minikit. Then gene expression would be quantified using nanostring and analyzed using NSolver 4.0 program. Results could be shown in heat map, gene ontology, etc., etc. Nanostring and counter gene expression system technology can gauge and assess specific gene expressions which come in forms of a variety of gene panels. A gene panel can contain 750 to 800 genes. It is an amplification-free technique, which means that there's no need for polymerase, but rather utilize fluorescence color-coded barcode-labeled probes to hybridize to the selected nucleic acids, for example RNA in this case, and adding up fluorescence signals to determine the amount without conversion to cDNA and amplification. This can reduce the amount of hands-on time. The traditional RNA sequencing requires conversion of RNA to cDNAs, then cDNAs are amplified and quantitated. This technique can contain variability which results in bias and may be unable to replicate the results. Main steps of the technique are as follows. Firstly, hybridize the RNA to two probes. One is reporter probe, which is tagged with fluorescence, and capture probe, which will be attached to this cartridge. The excess probes are removed, leaving only purified target probe complexes. After purification, these complexes are attached and aligned to the imaging surface. The sample is then scanned by a microscope where the fluorescence labels are counted and analyzed via software. This is an example of a heat map. A heat map is a data visualization technique that shows magnitude of a phenomenon as color in 2 dimensions. This is an example of a gene ontology enrichment analysis. One of the main uses of the gene ontology is to perform enrichment analysis on gene sets. For example, given a set of genes that are upregulated under certain conditions, an enrichment analysis will find which gene ontology terms are overrepresented using annotations for that gene set. In summary, this project aims to identify and analyze gene expression of MDSCs in the knee joints of osteoarthritic patients to further understand the pathogenesis of osteoarthritis. Thank you. For the last question, in which kind of diseases can MDSCs be found and what is the function of these cells?